Okay, it's a great pleasure to be here, and thank you for inviting me. I'm from the extreme biological end of molecular biology, so quite a shock for you. And until I started working on cell movement about 10 years ago, I hadn't had a physical thought since I left school. I mean, it's absolutely not necessary to be a molecular biologist and think in terms of physics. And of course, if you haven't done it for 40 years, it's a struggle. So I have two stories to tell you. One is about chemotaxis and an attempt to identify the core proteins that drive chemotaxis. And the second is a new departure for my lab, which is actually going to be our final flourish, which is how cells drink, how they take in large volumes of fluid through cup-shaped projections called macropinosomes. And I've sort of devoted the central theme of my career really has been to use dictyostelium, a soil amoeba, and exploit it in different ways to try and answer fundamental biological problems. And I thought for this audience it might be fun and useful for you if I just described a bit of the biology first. I don't know if that would be useful or not, but I'll do that. And then these two problems. Okay, and I work out on the other side of town in this lovely new lab here. This field is now has been dug up and is now becoming the AstraZeneca headquarters. They're entirely decorative. I assume they're from the fume cupboards. Yeah. You have to have them a certain height, and I guess they've added these meshes around them for decoration. Yeah, this is all the air handling is done in these places here, so it's all separate from the rest of the building. Doesn't cause any vibrations. And if you get a chance, it's the most spectacular building to see. So, as I said, dictyostelium is a soil amoeba, and in case you want to know the phylogenetics of it, here's a tree of the eukaryotic life with humans and metazoa out here, the fungi here, the plants over here, and dictyostelium and other amoebae here. So you can see in the trunk going up to humans, it branched off before fungi. But in actual fact, it shares a lot of properties, similar properties with human cells, including the way it moves and the way it takes in material through macropinocytosis and phagocytosis. And that's probably because, pardon me, the last common ancestor of dicty and human probably had already invented those abilities, and they've been conserved with variation ever since. So this amoeba lives in the soil and eats bacteria, just engulfs them, eats them up, and grows. And while it's doing that, it's basically an asocial cell. So it grows separately, it takes little notice of other cells around. And the thing that attracted people to the organism 50 or 60 years ago was the fact that it appears to be on the edge between multicellular and unicellular life. So when these cells are starved, they aggregate together and make mounds of maybe 100,000 cells, of which this is one, which will eventually give rise to a fruiting body consisting of stalk cells and spores. So there's a sort of problem of how cells make decisions to become either a stalk cell or a spore, how they regulate that decision to get the right proportions, how those cells are arranged in space to get the right structure. And actually that's what I worked on for a long time. The early stages of development, where the cells are all separate, are driven by chemotaxis. So what happens during this stage is that cells release a chemoattractant, cyclic AMP, and move towards the sources of cyclic AMP. At the same time, when they perceive cyclic AMP, they release more of it, and this diffuses out to more distant cells. And so a whole field of cells will gather together into a mound, driven by chemotaxis and relay of cyclic AMP. And I'm going to show you next, which is nothing to do with my talk, but just pure fun. A movie that we made for the BBC 30 years ago now. And this is at the later stages of development. Oh, sorry, I'm not fully in control of my things here. Right. So these are multicellular aggregates or slugs, 100,000 cells migrating over an agar surface. So this is midway through the life cycle. And these guys are phototactic, thermotactic. They also can sense gases. And what they're basically doing is migrating through the matrices of the soil, trying to find a suitable place to fruit. And now one of those slugs is fruiting. And here's the stalk being laid down. These are the pre-spores that are going to become the spores. So here you have lots of problems of cell movement. You've gone from single cells chemotaxing to cells moving as a group. And there's interesting problems there, which Case actually absolutely pioneered, of how the cell movement is organized to get this sort of structure. And we're back to the beginning again. Okay, so I won't distract you with that for a minute. So the organism has about 12,000 genes. That's a sort of, scope that we've got to play with. 12,000 different proteins, each doing lots of different things, able to interact with lots of other things. Not at all simple. So this is chemotaxis of individual cells. What you can't see is this here represents a microneedle that contains cyclic MP, which is being released into the medium. And these cells have a marker for F-actin, so polymerized actin. And you can see that they very efficiently chase the needle. So this is the start of the movie. As they get to it, we tease them by moving the needle. They change direction. And there's a real problem here of how cells sense gradients of chemoattractant and how they organize their movement machinery to move. And I don't really want to talk too much about that, just a little bit. So that's the problem. How do the cells do that? The first point I want to make is, you can see here they're making classical pseudopods. So these are actin-driven projections where actin polymerizing underneath the membrane, making a dendritic mesh, pushes the membrane outwards. And a very good marker for this sort of behavior is the recruitment of the scar wave complex. So this is a complex that activates the ARP2/3 complex that you've heard about already, which in turn initiates actin polymerization. And basically where you see this complex recruited, there you see actin polymerization, and there you see extension of the membrane. And so these things are classical pseudopods. And one thing that we found is that cells have, dictyostelium cells and most other sorts of motile cells have at least one other alternative way of moving, which is instead of using pressure produced by actin polymerization under the membrane, they use fluid pressure to produce blebs. And in dictyostelium, this behavior is provoked if you face the cells with mechanical resistance. So this is now a cell moving under agarose. So it's on a glass surface, and there's agarose over it. And unlike collagen, this is a very fine grain matrix. So the cell is experiencing, probably I've forgotten now, I think the pores are less than half a micron. So it's meeting fairly continuous resistance. And what you can see is that it's forming blebs. So here again we have a marker for F-actin, And what happens is that there's a layer of F-actin under the cell membrane, and for reasons unknown, those become detached. And in that area, fluid pressure drives out the membrane very quickly. And then almost immediately, the cell rebuilds an actin matrix. And the previous matrix decays. And so these scars here that you see are the previous matrices. And if you look at the leading edge, you can see very rapid extensions. So this mode of bleb-driven movement, which we worked on with Till, can be entirely induced by resistance, just increasing the agarose, the percentage of agarose that the cells have to deform, switches them almost entirely from using actin-driven processes to using blebs. And recently we've made a cell squasher where we can put cells under low percentage agarose, apply a little bit of force, and immediately switch them to bleb motility. And the sorts of forces you need are of the order of 100 pascals, sort of pressures you need about 100 pascals to do that. Okay, so the other point I wanted to make about both pseudopods and blebs is that they can be orientated by chemotactic gradients, and this is shown here. So that this is actually in the same cells moving in the same conditions. Here you have pseudopods. Here's a gradient of cyclic AMP, and you can see that the cell orientates its pseudopods up gradient very efficiently. Blebs, it does something similar, but as you can see, it does it in two modes, either side of the true direction. So when you think about chemotaxis, you have to think about signaling going from the cell surface to at least two different forms of projection, both of which can be orientated by the chemotactic gradient. And 15 years ago now, Peter Devriotes and co-workers did this most beautiful experiment. So the basic idea that came from this observation was that cells have within them something like a chemotactic compass. This is what it was called, which is an internal representation. What they've done is to take the gradient, which may differ by only a few percent between their front and their back, compare the concentration of chemoattractant, and decide which direction has the highest concentration. And then they've made an internal representation of that, like a compass needle, which then organizes their movement. And what Devriotes found was that there's a phospholipid, so a small signaling molecule in the plasma membrane, which can be orientated by external gradients in this way, and this is PIP3. So in this experiment, actually, you have paralyzed cells with a reporter for PIP3, and again, a needle releasing chemoattractant and some dye, so you can see where it is. And you can see that all of the cells have made an arc of PIP3 towards the needle. It's a very striking experiment. My own personal belief now is that it's almost irrelevant to chemotaxis. And one of the reasons for that is the following experiment that we did. Well, I should just say something first as a comment on their original results. These are no longer paralyzed cells, but it's the same reporter. And although PIP3 can be found at the leading edge, it's much more commonly found in objects that become macropinosomes, become cups. So this here, for instance, would be PIP3, and you can see it's forming a cup-shaped structure. Here is a section through an extending cup. So most often, even in the first papers on this phenomenon, PIP3 is associated with macropinosomes, not with leading edges. So,
I mean, maybe it's more general, it's just collateralizing the actin.
Protruding lamella, right? Which what goes over the skin of sizes to protruding lamella, right?
there's a lot of actin in lots of places in the cell where you don't have PIP3. And I'll come back to something like that later, if I may.
What I mean is the newly polymerizing.
Sorry?
Because it's downstream.
Yeah, I'll come back to that. I think I sort of doubt that. Yeah, sorry.
If you were to look at PIP3 dynamically, would you see, as the introduction of the needle, would you see that spatial correlation with PIP3 expression that might help to address whether?
Okay, I mean, this is getting complicated. So I'll just say this as an aside. So all of this work is done with what we call axenic cells. And those had, until recently, an unknown mutation in them that allowed that upregulated macropinocytosis. That mutation we identified as a NF1, so it's a RAS gap. And all of these cells have activated RAS signaling and activated PI3 kinase signaling. And if you look in true wild-type cells, then you basically don't see PIP3 orientated with protrusions. However, the standard PIP3 reporter used is relatively insensitive. So this is a reporter called crack-PH. There are more sensitive reporters, and with those, you can see shallow gradients from front to back in wild-type cells that are not bounded by the pseudopods. So they will go continuously the length of the cell, but their highest points generally is in the pseudopod. And I think if there is a role of PIP3, it's likely to be in that sort of thing, giving an overall polarity to the cell, not specifying where it forms a pseudopod. Okay, so I will proceed with our experiment of 7 years ago, I think now. And so PIP3 is made by PI3 kinases, and the ones that are relevant are those that can be activated by RAS. So they have a RAS binding domain in them. And there are 5 of those in the dictyostelium genome. So we just did the bloody-minded experiment of knocking them all out, removing the ability of cells to make PIP3 under RAS regulation and to make gradients of PIP3 in the membrane. And there they are, all knocked out. And then if you look at the chemotaxis of these cells towards the needle releasing cyclase MP, you can see they do perfectly well. So this proves, I think, as a minimum that this PIP3 made by this set of proteins, and it is we've shown subsequently this is 90% of the PIP3 in the cell. There is still another source of PIP3 coming through another type of PI3 kinase, but there's no evidence that this can be regulated by receptors, which you need to do if you're going to chemotax. But with that caveat, this proves that PIP3 is not essential for chemotaxis. The cells do almost indistinguishably well. And there's been a lot of, I would say, groveling in the field ever since then, saying, okay, there are multiple parallel pathways, and PIP3 is one of them, and we have to find the others. And the part of that I agree with is that we actually probably don't know what the relevant chemotactic pathways are very well. And so the first thing I want to talk about of new results is our attempts to find out what are the key proteins in chemotaxis. Now, for somebody like me, the classic approach, which has worked endlessly in biology, is to make mutants. Do genetic screens, find mutants that are messed up in chemotaxis, identify the genes, do various other genetic tricks to put them in pathways, and then gradually study them individually, put that together, and then at about that stage, hand over to you guys and start making models to see if we actually understand it. So this is the traditional approach, is do genetic screens. It's been a little bit difficult. The leading organism for doing that in chemotaxis is dictyostelium, and it's been a little bit difficult because of 3 reasons, really. One is that we can't handle essential genes very well. Those are genes that are essential for the cell to live. You need to do clever genetics to handle those, and we don't have such clever genetics generally. The second reason is redundancy. So if you have 2 or 3 proteins doing much the same thing, then if you knock out just one of them, you tend not to see a phenotype because the others are taking over the role. And the third is something sort of mimicry. So the chemotaxis that we study occurs after about 5 hours of development. And in those 5 hours, the cells gain many of the abilities they need to chemotax, including the receptor for cyclic AMP, which is a typical G protein coupled receptor, and so on. If the cells don't develop properly, they don't get those things and they don't chemotax. So you're easily fooled by mutants affected in development. And as it turns out, some of the mutants that genuinely are involved, some of the genes that genuinely are involved in chemotaxis also regulate development. So it's difficult to disentangle. So we've taken a sort of more biochemical approach, and this is based on the assumption, well, it's not really an assumption, the fact that most proteins are regulated by protein phosphorylation. So the idea is that if you could find proteins that become phosphorylated in response to stimulating cells with chemoattractant, you would have a list of suspects of the proteins involved in chemotaxis. And this has become possible on a mass scale, on a sort of whole proteome scale, by mass spec and using SILAC, which labeling the cell. So what you do here is to construct an internal standard which you compare with your stimulated cells, and the internal standard is marked by the cells having been grown on heavy amino acids. And so on the mass spec you will find pairs of peptides, one from the control and one, say, 2 mass units heavier from the cells grown on heavy amino acids. And you can enrich for phosphorylated peptides and from their sequences identify the proteins. And so in one extensive mass spec run, lasting maybe a week or something like that, you can look at a quarter or a third of the total proteins in the cell. So we've done that. This was the basic setup for cyclic MP. So we did a very simple time course. We stimulated cells for 10 seconds, which is the fastest they will respond, 45 seconds, which is when basically all of the responses are present, and then 360 seconds to look for very persistent responses. So many responses come up and go down again. They adapt. These are non-adapting. And I should say that if you were to do a needle experiment, so have a cell, bring a needle, look how quickly it can orientate, then it will be producing a pseudopod, I'm guessing, in about 30 seconds. So we're spanning the sort of time that cells can respond to a chemoattractant. And so we've been able to find about 12,000 phosphorylation sites on about 3,000 proteins. We find both phosphorylations and dephosphorylations, and up to some very big changes, 150-fold increases in phosphorylation. And for our purposes, we've treated things as interesting if there's at least a 2-fold increase. We haven't really worked on the decreases, and of course, statistically significant, but I think biologically significant is more important, and that's at least 2-fold for me. So we have, we have about 300 proteins which respond to cyclic AMP in this way. And so that's a filthy big list. So we try to simplify it. And so what I should say is, so we've looked at developed cells, we've stimulated them with cyclic AMP, and we've got this set of proteins here. Now amongst those are proteins that control chemotaxis, but also amongst them are things that do other things, like driving cell differentiation, relaying a cyclic AMP signal to other cells, and so on. So one way of finding core chemotaxis proteins then is to take a different chemoattractant, and we've used folic acid. So this is used by growing cells to hunt bacteria. Bacteria inadvertently release folic acid, and this is how cells know of their presence. And although there's 5 hours starvation between these cells, but actually they're quite different in their properties. So we're looking at a different chemoattractant in quite a different proteomic background. And from that we get another set of phosphoproteins and an overlap. And that overlap is now reduced to 78, what I'm going to call core chemotaxis proteins. It's still a long list, and one needs to try and make sense of it. So one way we've done that is to look at the sites phosphorylated. So these phosphorylations we're concerned with always happen on a threonine or a serine. And part of the specificity for the protein kinase that does the phosphorylation comes from the surrounding amino acids. So you can look at those, and one thing you can see is this particular motif here, a serine, proline, arginine, or equivalent. This is the phosphorylation site, a threonine, proline, arginine. And we found that in this core set of proteins, there is more than half of them, 52 out of the 78, have this single motif phosphorylated. So that might imply that there's a single master protein kinase doing this whole lot. At this point, it becomes difficult because you can't, from this sort of consensus, predict what the protein kinase is going to be. You have to go and experimentally find it. So the way we set about that was, first of all, to track down an antibody that would recognize this phosphorylation motif. And the reason for doing that is that you can then do Western blots, where you blot the protein and stain it with the antibody and recognize proteins that have this phosphorylation at this site. And you can do this for many samples in one day, whereas it takes you weeks to do a single mass spec experiment. So here we have such an antibody, and you can see that if you stimulate cells with cyclic MP, then over the course of 120 seconds, these are phosphoproteins here, not identified. You can see that after a delay, you get strong phosphorylation. Okay, so we then use that as a tool, and this is not so relevant. The same is true of folate as well. So we then use this as a tool to go through mutants blocked in various signal transduction pathways and see whether this phosphorylation response depended on the particular protein knocked out in the mutant. And here's a sort of conventional, very simplified idea of the signal transduction that's occurring in chemotaxis. So basically it goes through a G protein coupled receptor, the G protein, and then a lot of other stuff. And we just looked in various mutants. So here you see a double mutant where we've knocked out these two things, and you can see that the response is intact. Here we've looked at another mutant where we've knocked out the ability of cells to make PIP2 largely. So this is a PI4P5 kinase. The response is largely intact. And so we say that the response doesn't depend on those components, and we carry on, and we basically knock out the entire pathway one by one, and we get the response remains intact. So you probably start worrying at this point. Are you even playing in the same field as you ought to be? So reassuringly, we can knock out the G protein right at the top of the signal transduction pathway, and there you can see the response is abolished. Finally, we found that the protein kinase, ERK-B, so this is a MAP kinase, highly conserved protein, when you knock that out, the response is completely abolished. So if you knock out ERK-B, then cells no longer phosphorylate this core set of proteins in response to either folate or cyclic MP. And to confirm that, we then redid the mass spec experiment, and these are the substrates for ERK-B, and you can see instead of being on the diagonal, they're now along the x-axis. Pardon me. Okay, so what I think we've proven is that a major part of the core proteins, core chemotaxis proteins, are phosphorylated, regulated by a single protein kinase, ERK-B. And then, I hope I'm not going to run out of time, but the question is, what is it doing? And here, we just don't know. But we have a sort of working idea. It's not even a hypothesis, I'd say. And what we think it's to do with is adaptation. So this is the ability of cells to adapt to different ambient concentrations of their chemoattractant and still chemotax. So this cell here is perfectly happy in a gradient at low concentrations of cyclic MP because there's a big difference in concentration between the front and the back. If you then put it with a high ambient concentration, it's now only got a 2% difference between the front and the back, but yet it can still chemotax. So it's believed that there's some sort of mechanism to offset the ambient concentrations of chemoattractant, and probably, as you know, you can use that same sort of mechanism to actually distinguish the front and the back of the cell as well, so it can be continuously operative. And this is an example of adaptation from Peter Debriotes, slightly different. So one gives cells step stimuli of cyclic MP, and for each step you see a response that dies down, that you then increase the cyclic MP concentration, again you see a response that dies down, and so on, right up to where you've saturated the receptors. And the way this is explained in a formal sense is to imagine you have two responses triggered by receptor occupancy, one fast and one slower, and that this is an activating response, an activating signal for the response, and this is an inhibitory one. And only in the period where the activator is greater than the inhibitor do you actually get your response. And Debriotes calls this something and this anything, which is probably right. A concrete example would be something like this, which would be an incoherent feed-forward pathway where you have an activating GEF and an inactivating GAP, which work on a small G protein, and you would assume that this becomes activated faster than this, but eventually they equalize. Okay, so we're thinking that it's somehow something to do with that sort of process. If that's the case, the minimum we would expect is that the mutants in ERK-B should be badly affected in chemotaxis. Sorry, these seem to be alternative. I can do one or the other. No, both. So here you see wild-type cells chemotaxing to a needle-releasing folic acid. It's starting, they're going in. They're not brilliant at it, and there are reasons for that. Here's the mutant, much worse. Okay, so if one's going to affect an adaptive circuit, one of the other properties you expect is that although the output itself adapts, the circuit itself, both the inhibitory and the activatory loops, should remain active in the presence of the stimulus. Otherwise, the cell can't sense the stimulus. And this is shown by the fact that the phosphorylations that are mediated by ERK-B themselves do not adapt. So you can maintain a stimulus for as long as you like, and those phosphorylations remain. So unlike many of the outputs which are transitory, These are long-term responses. And this is shown here. This is arranged, so cells are stimulated with cyclic MP. This is arranged with a low dose, which is hydrolyzed by the cells over time and runs out. And when it goes, the phosphorylations disappear. This is a high dose, doesn't run out, and the phosphorylation just persists. So that's one, a second property you would expect of something involved in adaptation, that it itself should not adapt.
Well, can I just ask you,
Yeah.
with these stimuli, are you also stimulating proliferation, thinking of Earth's role in proliferation as well as?
In this case, probably not. The ERK-B has no growth defect. The ERK-B null has no growth defect. So, and these are starving cells. So, and also it's very quick. I mean, we're dealing in seconds here, not minutes or hours, which are involved in proliferation. Are you asking, could some of these substrates be involved in proliferation? I would say not, because of the way we've got them as an overlap between two chemoattractants, one of which has nothing to do with proliferation, I would say. I mean, I'm going to leave you very hand-wavy because I really don't know what's going on here. But just to go back to our concrete example, so here's of what we think ERK-B is doing. Here's our incoherent feedback loop, and what I'm proposing is that ERK-B takes out both sides of the loop, so that you reduce. I was trying to think this morning because I sort of had a concrete idea which I've forgotten by this morning. But you actually reduce the active number of both the inhibitory and activating components in the loop. And that will allow you, I think, to adapt to higher concentrations of chemoattractant. Okay. So just to summarize, what we've done is to identify, I believe, a core set of chemotaxis proteins. And there are similar experiments going on with HL60 cells, and we're going to see if this is a universally conserved core set of chemotaxis proteins. I didn't say anything about them, but they're exactly the sort of things you would expect. They're GEFs and GAPs. They're regulatory proteins. So GEFs, GAPs, protein kinases, PI kinases. I didn't also mention there's quite a lot of internal validation. So null mutants existed already for 30 of the proteins that we identified, some in very different contexts. And of those, 24 have or are likely to have a chemotactic defect. And that of this set of conserved proteins, one protein kinase, ERK-B, mediates the phosphorylation of more than half of them. And we're proposing that ERK-B is something to do with adaptation. Okay, now I'm going to change to the other project that's exciting us at the moment. And this is about macropinocytosis, and I'm going to present it slightly backwards. So macropinocytosis is the uptake of large volumes of fluid from the medium into vesicles of 2 or 3 microns in diameter, so much bigger than through clathrin-mediated endocytosis. And it's used by dictyostelium cells for feeding. It's also become apparent recently that it's used by cancer cells, RAS-activated cancer cells, for feeding as well. And it's important in the immune system for antigen processing. And it's probably important in quite a lot of other ways. It's a very mysterious cell biological process. Very little worked on, actually. But it appears to be highly conserved. And I wanted to show you this lovely photograph. This is all the work of one person, Douwe Veltman. And he gave me a very grim-looking photograph of him being very serious, and I wanted one of him smiling as he normally does, and here it is. So this is Douwe's work, and Douwe is a combination of tremendous microscopist with ability to analyze images and also a very good molecular biologist. And this is a really rare combination, so he can make lots of reporters. And what he found was this beautiful image. And this is the answer. But the question is, what was the question? And I'm afraid this is the way at least our science goes quite often. So there's the answer. What on earth was the question? So just to give you some clues, it is a cell. This whole thing here is one cell. The red is PIP3, which we've already talked about. So this red here. And the green is the scar wave complex, which activates actin polymerization, as we've talked about. Okay. And so what this cell is doing is macropinocytosis. And here is a movie of living cells doing macropinocytosis of dextrans, so fluorescent they're in a fluorescent medium. And here you can see carry on. You can see these ghostly projections going out and trapping a droplet of fluid which is taken into the cell. So that's macropinocytosis. Here is the most beautiful movie. This is made at Genelia Farm using a Bessel beam microscope. And on the left are the microtubules, which I'm afraid for these purposes you can ignore. And on the right is a marker for polymerized actin, F-actin. And you can see what the cell is doing is producing cups, which at a certain point close. And those are macropinosomes. This is just incredibly gorgeous, I think. So, the question becomes clearer. So, macropinosomes and also phagosomes and also circular ruffles are cup-shaped projections formed by cells, and they're formed by a ring of actin polymerization. And those rings can be several microns in diameter. And so the question is, how the hell can a cell organize a ring of actin polymerization several microns in diameter? It's not polymerizing actin in the middle, just in a ring. So in the case of the much smaller vesicles taken in by clathrin-mediated endocytosis, you have a sort of crystallization process that produces the vesicle of actin and its coat proteins, and it's sort of easy to understand how you can get a cup-shaped structure in that situation. When cells take in large particles by phagocytosis, when they take in yeast, what's believed is that the particle itself acts as a template for the formation of a cup. The cell senses the particle and just polymerizes along it in what's called a zipper process. But for a macropinosome, there's nothing like that. There's no known, of course there are structural elements, but there's nothing like a coat known. And there's no solid particle to organize it. So what is the template for making a ring of actin polymerization? And I think what we saw in the first slide was the answer to that. So just to try and start quantitating this, first thing that Dower did was to look at cells expressing a marker for the scar wave complex, which is in green here, surface render the cells so that he could recognize macropinosomes, then take the rims of them and compare the intensity of scar wave staining there to the cup. So I should just, as a sort of aside, say that what we've done is to put quite a large protein on quite a small protein that goes in a complex that forms a complex with another complex, and you worry that your reporter is active or not. And this is evidence that the reporter we're using is active. So we turn to pseudopods, and here's the reporter, and here's a chymograph showing that where the reporter is present, then you get movement of the leading edge here. Where it's absent, you don't. It's present, you get movement. Absent, you don't. Or you can correlate presence of reporter with movement. And both these arguments suggest that the reporter is reporting active scar wave complex, which is triggering actin polymerization. Okay. So, as I said, we try to first of all quantitate the recruitment of both PIP3 reporter and of the scar wave reporter in recognizable macropinosomes. And basically here's a profile across them, around the macropinosome, I think, I'm not quite sure, where you can see that the PIP3 reporter is in the middle, and either edge there's a rather faint and often difficult to recognize and often sporadic recruitment of the scar wave reporter. And when you look at a number of macropinosomes, the key point is that in the center you obviously have PIP3. By definition, the way we've done it, at the edges you don't, but you see in the center you don't have scar wave recruited, and at the rim you do. Okay, so that's the first point. We then got this idea that basically if you see a PIP3 patch, then what's going to happen is you're going to see scar wave recruited to the edge of it, but not to the center. And so we attempted to generalize that by looking at all sorts of PIP3 patches. So, and these can be produced in relatively arbitrary ways, but if this is a general rule, it ought to be true in all cases. So one way of producing, well, the first way we looked at is just in sections of cells, so single confocal sections. You see PIP3 patches. Sometimes these are clearly associated with macropinosomes, but not always. But you take those, whatever they look like, and you do the same measurement, and again you see that in the center of a PIP3 patch you don't get scar wave recruited, and at the edge you do. You can induce acute patchiness by stimulating cells with cyclic AMP, and this is shown here. So here at T0 cyclic AMP is added to cells. We're looking at a reporter for PIP3, and initially you get a global recruitment to pretty much the whole plasma membrane, and then it resolves into a number of patches, and often these visibly are macropinosomes. If you look at the scar wave reporter, it's patchy, but if you look in a chymograph, pardon me, it's always excluded from it's around the edges of the PIP3 patches. Here's a thing for TIL. So on the base of axenic cells, these mutant cells, you see very dramatic and beautiful what Gerish calls actin waves, but actually what they are, I believe, is PIP3 patches surrounded on all sides by actin polymerization. And so here you see here you see pH crack, here you see scar, and here's the overlay, and it's very clear. If you look at other activators of the ARP2/3 complex, the WASP in particular, you also see rings, but less clear. WASH you do not. So basal waves, it's the same story, where you see a PIP3 patch, you see scar recruited to the edge, not the middle. Sometimes during the later stages of aggregation, cells will form head-to-tail streams, where they hold the front of one cell will latch onto the back of the cell in front of it. Here's a stream of such cells, and what you see is that the front cell has a patch of PIP3, which is changed color, it's orange now, and around it you have scar wave, and here's a 3D reconstruction. So again, the rule is applying. And finally, in phagocytosis, where you're taking up a solid particle, and actually we found this the hardest of the ones to image, but and this is the best we can do, and you can't see the particle, but this cell here is taking in an invisible yeast here. And there's a patch of PIP3, and around the rim there's recruitment of scar wave. So in all the circumstances we've looked at, this appears to be a rule of the organisation of the dictyostelium cytoskeleton, that PIP3 patches repel scar wave from the body and attract it to the edges. So just to go back and give a genetic test, does this mean anything? Here we go back to the PI3 kinase mutants, which were largely unaffected in chemotaxis. If you look at their ability to take up fluid, then, and they're taking up phytsidextran here, here's the wild type, takes it up efficiently until it starts, and then starts releasing it again, so you get to a steady state. The PI3 kinase multi-knockout here takes up barely any, and this is probably by other routes. And you can see this here. This is the fluorescent fluid inside the cell, and here's a mutant where only 2 of the kinases have been knocked out. So genetically, PIP3 is very important for chemotaxis. That isn't the end of the story at all, though. As I said, this class 1 PI3 kinase is controlled by RAS. And so then the question arises, what's the role of RAS in this? And what we find is that PIP3 patches are invariably supported by a patch of activated RAS. So again, there are reporters for the activated state of RAS, and here one's looking at a basal patch, and this is PIP3, and this is RAS, and this is the overlay. And if you compare it pixel by pixel, then you find that where there is RAS, I didn't emphasize this earlier, but pixel by pixel, where you see high PIP3, you don't see scar wave, but pixel by pixel, where you see high PIP3, you see high activated RAS. So PIP3 is important, but the PIP3 patches are supported by patches of activated RAS, and you can then ask various questions about that. For instance, could the patches themselves be formed by a feedback process where PIP3 feeds back onto activation of RAS, and you get they're dependent on each other. And you can ask questions about that, again, by looking at the null mutants. And the short answer is that the RAS patches are independent of the PIP3 patches. So you can knock out the ability of the cell to make PIP3, it will still make RAS patches, and those RAS patches will still, I hope I'm looking at the right thing here, they will still recruit scar wave to their edges, but less efficiently than if PIP3 was also present. And this is shown quantitatively here. Okay, I'm going to just summarize that, and we can speculate about how it might happen. So the summary of this part is that it seems to be a rule of the cytoskeleton in dictyostelium, but I think it's going to be general this, that scar wave is attracted to the edges of PIP3 active RAS patches and repelled from their centers. And of course, this automatically, if you can make a patch and you have this rule, you can make a ring of actin polymerization. So this gives a template for cells to form cup-shaped structures. And so we're proposing that this is a common route for forming macropinosomes, phagosomes, and circular ruffles. A very ancient trick that the cell has used. First, create an activated patch of RAS PIP3, have a mechanism for recruiting scar to the edge, trigger actin polymerization there, and you've got a cup. Okay, and this is the group at the LMB, as it was a year or two ago. So here's Dower. This is sort of his work face. This is John Nichols, who did the Silac experiment. Yevgeny Zachilevsky worked on blebs. Gareth has done lots of genomics and genetics. David is my long-term associate, David Traynor. This is our new PhD student, Tom Williams. And thank you very much.
Questions for So you initiate the cup, how do you close it?
Yes. As I said, it's not a well understood process. My impression from looking at the Bessel beam movies is that there's an active process there. It looks to me as if the cup grows, and at a certain point something happens and then the rim contracts quite sharply. If that's the case, it would suggest that there might be myosin 2 involved. And one could obviously work through the myosin mutants, which we're starting to do, to see if there are deficits in closure. But I mean, I think the mysteries of this are how you form rings, how you close cups, and how the membrane fuses. And I don't really know about that.
So you said in phagocytosis, it is the particle that directs the formation of the ring. So is macropinocytosis perhaps the very same thing, but with a randomly selected location of your initial PIP3 scar recruitment?
I do think they're very, very, very similar processes. And I probably would say that the particle directs the formation of a ring by activating signaling that activates RAS, produces a RAS patch which is restricted to the contact between the particle and the cell, and then the automatic recruitment rule kicks in and you get scar recruited to the edge. I don't know if that's restating what you said, but
So the downstream effectors should be
yes.
very same.
So I mean, we have some genetic evidence for that, but it's more complicated. So the size of the patches is controlled by this NF1 RAS gap. So if you have active NF1, then you have small patches, and your macropinosomes are small. And it turns out you can't take in large particles. So you can't take in 3 micron particles, yeast-sized particles, very well. If you knock out NF1, you get bigger patches, you get bigger macropinosomes, and you can take in bigger particles.
Does curvature matter at all? The curvature, the shape of, does it matter? I mean, do you, if you didn't have these sharp edges, would you get the rings forming?
Well, I mean, to get a ring, you've got to both have a ring of actin polymerization of some width, but you've also got to have not actin polymerization in the middle, otherwise you'll get a cylinder. So that's one thing that's necessary. But then you're going to have a lot of membrane tension, where you go up and down again, and you're going to have to anchor the membrane quite carefully in the middle of the ring, and you may need to have assistance in bending the membrane, especially I think at the lip. So I think, there's a lot more to it than just these guys.
When the cup grows, is the inside still covered with PIP3 and RAS?
Yes.
So that goes all the way?
Yes, so it's basically, they're restricted up to the rim. And unless the cup grows enormously, which you actually see in basal waves, which I regard as equivalent, basal waves on the bottom of cells, when they get too big, then you can get the center hollowing out. And actually, if you look at the profile of a PIP3 reporter, then what you see is it goes up, plateaus, and then goes down quite sharply over a course of half width of a micron or less. But I think if that central saturated area gets too big, then you can't sustain the patch and you get a hollowed out thing.
In the back. In the back. Sorry, with respect to the geometry, is there any similarity to stomatocytes?
If I knew what a stomatocyte was, I would tell you. What is it?
It's like the vesicle forming, kind of like goes inside, so I can.
Is it on a similar scale to this, made by a single cell?
Yeah. Is that the same? Okay.
I'd like to know. I mean, you tell me afterwards what they are, and I'll try and find out.
Okay, in the main sequence of the red blood cells, so if you go away from the viscocyte of the red blood cell, you either go to stomacytes or to echinocytes.
I see.
It's one of the two directions.
I see.
Basically, if you change the spontaneous curvature of the system, you
Right. So that's not an actin-driven process.
No, it's driven by the membrane usually.
Yeah. So I think the answer would be that it's, at least in its mechanism, independent.
Last question. So, do you have an idea of when the cell is using this mechanism of macropinocytosis versus just, regulating its ionic composition through water proteins and things like that? Is there a competition that's going on, or when is this phenomenon really sort of important and taking place?
So basically, macropinocytosis plugs into an entire digestive system. So it's the cell's way of taking in stuff from the outside, digesting it, and extracting what it wants from it. So when these vesicles are taken into the cell, you then deliver lysosomal enzymes to them. You digest what's inside them. If it's a protein, if it's, say, in the case of phagocytosis, if it's a pathogen, you break it down. In the case of immune sampling, if it's a protein, you break it to peptides. In the case of dicty, you just break it down, and then the cell extracts, through mechanisms I don't know about, amino acids and whatever from the vesicle, and spits out what's left. So it's completely independent from water pour-ins and that sort of thing.
Okay, let's thank Rob again for and so we'll have to make